Well, hello, everyone. Welcome back. This week's lesson is about the muscular system. We're about one halfway done, so keep up with those quizzes on time and the assignments. Don't forget about your paper. You'll have to start writing it pretty soon. It's actually due, I think, the third week in November. I also need your topic if you didn't hand that in yet. Okay, for as far as muscular tissue goes. Um, there are three types of muscles in your body: striated, smooth, and cardiac. Striated muscle is also called voluntary because it moves when you decide to move it, except if you have a tic or something like that, or some involuntary movement. But 99% of the time, you move them on your own. They move your bones mostly, so they're also called skeletal. There's some striated muscles that don't move bones. For example, the ones that move your tongue and the ones that move your eyes around. But most of the time, they move a bone. They're long worm-like cells. They could be as long as one cell can be as long as the entire muscle. Very, very thin, though. You would still need a microscope to see it. They have lines on them. It almost looks like an earthworm with lines on his back. These lines, you'll see what they are later on in the recording. They're multinucleate, which means that because they're so long, it's better for them and more efficient for them to make their proteins using many nuclei instead of just one. Smooth muscles are involuntary. They still are controlled by the brain. They're called visceral sometimes because they are mostly involved with your internal organs. They're small cells. They have little points on the end. There's one nucleus in each one, and there's no lines that are visible, so that's why they call them smooth. They're the ones that churn the food around in your stomach and move it along in the intestines or cause your pulse to beat. Cardiac muscle is only found in heart. It is involuntary. It beats approximately once per second or a little faster on the average. You see some light lines on it. And they are branched. The cells are branched. They look like they have little arms coming out of them. They're uninucleate, meaning they have one nucleus, but they have a connection between cells that are different from the other muscles. These connections between the cells are called intercalated discs. And intercalated discs speed up the impulse across the heart from one muscle cell to the other, so that when the right side of the heart actually gets the impulse from the brain to beat, it speeds that impulse across the heart very quickly so that both sides of the heart beat at the same time. And the heart, of course, beats rhythmically. It's the only muscle also that being controlled by the brain. You can take the heart out of your body, not a good idea, but if you do it, the heart will continue to beat until it runs out of ATP. So there are three kinds of muscle cells. In the next slide here, we're going to talk about what the functions of muscles are. Now, obviously, they produce movement. They are able to move your bones so you can walk and talk and skip and jump and all of the things you do with your bones. They also maintain your posture as they pull against the bones one after the other, and they hold you kind of together and stabilize your joints. But also, when you contract a muscle, you generate heat. That's because in physics, whenever you use some energy, the energy is not really lost. It's just converted to heat, and the heat just leaves the system. So whenever you run those ATPs to contract a muscle or contract many muscles, you release the energy from the ATP. The ATP uses it to control the muscle and to contract the muscle, and then that energy is converted to heat. And that's why if you are working out very hard or you do something very strenuously, you get warm. So yeah, the muscles also generate heat. It's also why you shiver when you're cold. And it's not really to move a muscle or anything or move a bone, but also to just generate heat so you can warm up a bit. Now this little guy here is our chimp. He is very similar to humans, 99% basically the same inside. And so we can look at the muscles. The skeletal muscle there, that's the deltoid muscle. That's the one up in your shoulder that lifts your arm up. And you can look at the microscopic picture of that muscle, and you see that the cells are very long and worm-like. Those are all one cell each, and they have many nuclei in them. Those little pink little lines inside are the striations or lines that we'll talk about later on, which is why also they call them striated muscles. They're voluntary, and obviously the chimp uses it when he wants to raise his arm. The smooth muscles, you notice, are in his intestines here, and they look very, very different. They're involuntary. Obviously, you don't want to think about moving the food around inside of you. It's all done automatically, and also for pulse. The muscle cells are pointy on both ends. They're called spindle-shaped, and they have one nucleus each. And they're basically involuntary. They are still controlled by the brain, but you are not aware of it. The cardiac muscle is in your heart only. And if you look at the diagram there, you can see some of them show branches, one nucleus each, and there's still some lines in them. But they have those connections, those special connections between the cells, the intercalated discs. That speeds up the impulse across the heart. So both sides of the heart muscle beat simultaneously. You have a right and a left part of your heart, and they both need to beat together. And so in order to get that to work right, the impulse has to shoot across the heart faster than it normally does in a regular muscle. It's a sort of an electric shock, and that makes both sides of the heart beat at the same time. Now we'll talk about what that impulse is actually next week when we talk about the nerves. But for now, we'll just say that it's a nerve impulse and a muscle impulse, and just think of it as kind of a shock going across the heart or down a nerve cell. But next week, we'll talk about what it really is. In the next slide here, we're going to talk a little bit about smooth muscle. Just as a quick review here, you can see they look like little spindles pointing both ends. If you cut them in the middle and look at them from the edge, they look like circles. It would be like if you cut a pepperoni and then you look at the circles, the pepperoni is long and thin, but if you look at it from the edge, it's round. So you see the two different ways of looking at the muscle. No striations means no lines. That's why the smooth word is involved in its name. And spindle-shaped cells, one nucleus per cell, involuntary, and found mainly in the walls of hollow organs. And you see it here in the intestine wall. And you can see there's two layers of them, and situated in different directions. That gives the muscles layer of your intestines a little bit more strength, kind of like plywood. And the inside of that is all folded up epithelium tissue, which lines the inside of your intestine. The food will be going through the middle of that, and the smooth muscle, will be squeezing, allowing the food to move along. The next slide will show you the heart muscle, again found only in the heart. You see the branching, one nucleus per cell, and they have those intercalated discs to speed up the impulse across the heart. Its function obviously is to pump blood around the body. It is involuntary, and it's one nucleus per cell, and you see some striations or lines in them. Now this is where we're going to spend most of our time today. This is a voluntary muscle. You can think of it as your bicep, something like that. And we're going to look at the parts of the muscle now. Now in real life, this would be bright red because the covering of the muscle, called an epimysium, is actually connective tissue, which has lots of blood vessels in it. So you would see that as a red color. The periosteum is the connective tissue around the bone that the muscle is connected to. It's very tough, very dense connective tissue, hard to rip. And then it's continuous with the tendon of the muscle. So it's not like the muscle is glued to the bone. It's actually continuous with the covering. So it's very, very well attached, hard to rip off. That covering, that connective tissue covering, the dense connective tissue in the tendon, which is continuous with the periosteum of the bone, extends all the way around the muscle. And so it becomes the outer covering of the muscle. And that dense connective tissue on the outside would be called the epimysium. Remember, epi means on top of something, and my means middle. So it means on top of the muscle. Now, if you slice the muscle in the middle, you got that little belly of muscle piece sticking out there. you got a little piece you can look inside the muscle a little more clearly. Inside the muscle, it looks like there's little islands. Those islands of cells are called fascicles, which are really groups of muscle cells. The light gray that's surrounding the islands, that's called the perimysium. And peri, again, means around. And it's also dense connective tissue. A lot of blood vessels. That's why, muscles are red, and if you eat meat, it's basically muscle, so it looks red. And if you look at the islands have all these little circles. Those are cut muscle cells. They are surrounded by sort of a dark gray. And inside the island, there is an endomysium. In other words, a dark gray connective tissue, which is really red, surrounding each cell in the island or in the fascicle. So inside of a muscle, aside from the muscle cells, the most common tissue in there is connective tissue. And the reason for that is because when the muscles contract, you don't want, if you pick up something heavy, you don't want the muscle ripped. So you have lots and lots of connective tissue in the muscle, which is why there's so much protein in meat. That would be mostly collagen, elastin, and those fibers between the muscle cells, between the connective tissue cells. Now, inside of the islands, you see the muscle cells. Now, the muscle cells, again, are long and thin, almost like worms, but because they're cut, they look like circles. But you're pulling out one fascicle, and you can see the connective tissue around all the islands inside. That would be the endomysium inside the muscle. And one of the muscle cells is being pulled out. So you see how it looks. It actually is a worm-like cell, and it would extend all the way from one end of the muscle to the other end, one cell, tendon to tendon. Now, inside the muscle cell, which is sometimes called in books a muscle fiber, and I don't like that because it makes it sound like it's a piece of string or something. It's an actual living cell. And so it has all the cell parts. It's alive. It makes ATP and it's mitochondria. It does all the stuff a living cell is, all the genetics, everything. But we're going to call them in our course, we're going to call it a muscle cell because that's what it really is. But most books will call it a muscle fiber. So muscle fibers are muscle cells. Now, inside the muscle cells or inside the fibers, there are other fibers. That's why it's a little confusing. They call those myofibrils. Myofibrils are actually what contracts inside the muscle, and each muscle cell has many myofibrils inside, and you want to think of them like little rubber bands inside the muscle cell, which can contract and put them all together. If they all contract at the same time, the muscle can contract, and you can pick something up. So the myofibrils are the actual unit that works the muscle. They are little fibers inside the muscle cells, and we're going to take a microscopic look at those in the next slide. So let's do that now. And here it is. Now this is a slide where you have a muscle cell cut on each end, so it's only a partial muscle cell. And it's the red part there is the cell membrane, and we slice the cell membrane wide open. And what do you see inside? Well, you see 3 cylinders. Those cylinders are the myofibrils, one of which was sticking out of the muscle cell in the last slide. In a real cell, there'd be like 30 or 40 of those in there. Now, around them looks like all that gray stuff. That's the endoplasmic reticulum. Now, in muscle cells, they don't call it the endoplasmic reticulum. They call it the sarcoplasmic reticulum, because sarco is a prefix that refers to muscles as well. It refers to flesh, actually. And so what happens is that that's the sarcoplasmic reticulum, and we abbreviate that as SR instead of ER. But it's the same thing as a regular ER in any other cell. There's also what they call T-tubules, or membrane-bounded tubes, similar to the cell membrane, that actually extend from one side of the muscle cell to the other across it. And that would be called the transverse tubules or T-tubules. And you'll see that they have a function when we talk about the physiology in a few minutes of how muscle cells actually contract. There would be mitochondria in there, those things that look like little watermelons on the bottom there. Those are mitochondria. And you need a lot of mitochondria in muscle cells because, remember, the mitochondria do cellular respiration, which produces the cell's ATP from your food. And so that is where the energy comes from on the muscle here. There's also, all the other cell parts in there too, but there's nuclei, there's ribosomes, all of those things. But we're just talking about the parts here that are crucial to work the muscle. The myofibril, that's the key. Now you got 3 of them in there. They're yellowish in this picture. They're not really yellow. I just colored it to make it look like you can see them better. But if you take and look at the T-tubules, underneath each one of those, now you can't see it, but underneath each one of those is a wall. Each myofibril is broken up into sections, kind of like classrooms. And each section, which is bounded by a protein wall inside the myofibril, each section is called a sarcomere. And one myofibril, as it extends from one end of the muscle cell to the other end, can have thousands and thousands of sarcomeres. Remember, in this one, there's only 3, and because the muscle is cut. Now, in the diagrams below, I've taken away the sarcoplasmic reticulum so you can see what's inside of a sarcomere. Now you see the arrows, it's pointing to that middle sarcomere, the one in the middle of the myofibril. If you take away all the stuff on the top, what you see is the actual walls. The walls are called Z-lines. They're made out of protein, and they cause the myofibril to be broken up into many, many sections. And each one of them is called a sarcomere. The Z-line is made of protein. And if you look at the sarcomere, you see thin lines in there, and you see thick lines. The thin lines are a protein called actin, and the actin is actually attached to the Z lines. The thicker lines, the thicker ones, those are called myosin, and myosin is suspended in between the actin fibers. And the myosin has actually little looks like the feathers of an arrow on both ends. Those are called cross bridges, and they are cocked back. They are pulled back like a mousetrap, ready to spring. And right now they're just sitting there, so you can see the diagram. But in the next few minutes, you'll see how they work. And if you look at the closer picture, you'll see the is the big black line. The cross bridges are cocked back, toward the Z lines. And then if you look at the actin, you see something new. The little thin fibers that are attached to the Z lines are surrounded by what looks like a string of pearls. And what that is another set of protein molecules called tropomyosin. And the little dark ones there are called troponin. And they're around the axon. As long as they're there, the cross bridges, which are cocked back like a spring, cannot attach to the actin. This muscle cannot contract because the tropomyosin is actually in the way. What the muscle is going to try to do when it needs to contract is attach those cross bridges to the actin and then spring the trap and put the cross bridges into a different configuration toward the middle. And that, when they're attached to the actin, that'll pull the 2 Z lines closer together. And so it's like if your hands were stretched out and you're pulling on a rope, the rope will get, if it's attached to 2 things, those 2 things will get closer to you. The tropomyosin is the switch, really. As long as that's in the way, like it is in the diagram here, this muscle is relaxed. It cannot contract. It's ready to. The cross bridges are cocked back, ready to go, like the mousetrap is set. But they can't do anything because they can't attach to the actin. Literally because the tropomyosin proteins are actually in the way. So that's basically the coarse and the fine structure of muscles. The gross structure or coarse structure is what you saw on the last slide with all the different coverings, the epimysium, perimysium, endomysium inside the muscle, and the muscle cells themselves inside of fascicles, little islands called fascicles. That's the gross or coarse structure, which you really can see, with your eyes. The fine structure is when you look inside of a cell like this. You need a microscope to do it. And this is the anatomy of the cell itself. You need a microscope to see this stuff. And this is what's inside each of every one of your muscle cells. Now let's take a look at the next slide here. In the next slide, you see that from your nervous system, your spinal cord, there are nerves that come out and basically go to the muscle cells in your body. And you look at the muscle cells, you notice that there's two nerves there, a unit 1 and unit 2 coming out of the spinal cord. And each one of them is going to different muscle cells. One nerve can actually control more than one muscle cell. It looks like one of them, the blue one on the bottom there, is controlling 2, and the green one is controlling 3. And so those 3 or 2 muscle cells controlled by one nerve, the nerve and the cells combined, they call it a motor unit. So if you actually send a message down those nerves, down, let's say, motor unit number 2, it would only affect those 3 that it's attached to. And then motor unit the first one would go to the other 2 if you send the message down there. And that's one of the ways you control how much the muscle is contracting, how fast and how strong it's going to be. If you want to contract it very quickly and pick up something very heavy, you would send the message on all the nerves and control and make every one of the muscle cells contract. If you want to, just go slowly and not pick up something that's very heavy, you might not send messages to some of the nerves, and so some of the muscle cells won't get the signal, and not all of your muscle cells in your muscle will contract, and you'll get a weaker, reaction muscle, and it'll move a little slower. Remember, when a muscle cell does get a signal to contract, though, it's an all or nothing thing. It's kind of a rubber band boinging back. You can't do it halfway. And so every muscle either contracts or doesn't. And they call that an all or nothing response. Now we go to the next one here. This is a book diagram of a piece of a muscle cell, like the one you saw before, as a quick review here, showing it in a sort of a different way. This one has a muscle cell sliced in the middle, so you're not seeing the whole cell. And because it's sliced, you can see the myofibrils are sliced, and you see there's 1, 2, 3, 4, 5, 6, 7, 8, 9 of them in there. As I said, most muscle cells have about 30 of them. And then inside of the cells around all those myofibrils, you see lots of mitochondria. That makes sense, and they need a lot of energy. And then if you look at that, the blue stuff, that's the sarcoplasmic reticulum that surrounds all this stuff. And if you take that away, you see the sarcomeres. The blue lines are the Z lines. That's the walls between the sarcomeres. The pink, thicker fibers are the myosin with the cross bridges on them, but in this diagram you can't see the cross bridges. And the actin are the thinner ones. And so we'll see how they actually work now in the next part. We're going to talk about physiology now. Now, when a muscle contracts, this is what happens. You have to tell it to. Especially it's a voluntary, even an involuntary muscle is still controlled by your nervous system. When a muscle contracts, the actin and myosin filaments inside the sarcomere, they move over each other kind of like the prongs of a fork, of 2 forks. The way they do that is because the cross bridges on the myosin, which are cocked back like a mousetrap, attach to the actin, and then they boing. They do a power stroke, and they pull those Z lines closer together by grabbing onto the actin and pulling. Each one of the sarcomeres does that in a muscle. If you add it all up, you have some power, and you'd be able to do something with the muscle. That process of the filaments sliding over each other, they call that the sliding filament theory, and here's some of how it works. Now here's your diagram again, just to review. If you look at the bottom, you see the sarcomere. The sarcomere is bounded by Z lines. It's part of the myofibril. There's many sarcomeres in each myofibril. The dark lines are the myosin. The thinner lines, which are attached to the Z lines, are the actin. The cross bridges on the myosin can attach to that actin, if allowed to, and then boing to a different position. In other words, change their angle with power, and that will pull the 2 Z lines together, closer together. That is how the muscle contracts. If you add up every sarcomere doing that. The diagram next to it, you see the tropomyosin wrapped around the actin, and right now that muscle is unable to contract because the tropomyosin is in the those cross bridges cannot attach to the actin and pull because they just can't get to it because the tropomyosin is there. So what happens? When the action potential is the nerve impulse. You'll learn about that next week. Think of it sort of an electric shock going through the nerve for today. When the action potential reaches the muscle cell's endoplasmic or sarcoplasmic reticulum membranes, what happens is that there's a nerve that goes to these muscles, as you saw, but there's always a tiny little space between the end of the nerve cell and the muscle cell membrane. That space is very, very important. They call that a neuromuscular junction, which is really not a junction because it's a space, but that's what they call it, an NMJ. Sometimes it's called a synapse, but it's a space. And so when the nerve impulse reaches the end of the nerve cell, getting to the muscle, it stops there. It doesn't jump over the space. What happens there is that at the end of the muscle cell gives off a chemical into the space. That chemical is called a neurotransmitter. And in muscles and nerves, that neurotransmitter is a very important chemical called acetylcholine, or ACH. The acetylcholine will diffuse across that gap and attach to receptor proteins on the muscle cell membrane. When enough acetylcholine attaches to enough receptors, it starts the same electrical impulse that went through the nerve shooting through the muscle cell membrane. They call that a muscle impulse instead of a nerve impulse. As this electric shock for today, next week again you'll see what it really is. When it moves across the membrane, it gets to the T tubules. When it gets to T tubules, it shoots down into the sarcoplasmic reticulum. So that's what I meant by when the action potential or the muscle impulse now reaches the cell's endoplasmic reticulum. When it does that, it changes some shapes of some proteins on the membranes of that endoplasmic or sarcoplasmic reticulum, allowing calcium ions to leak out. Remember, we said that the ER or SR in this case can store things. It was one of its jobs when we talked about the cells. In this case, the sarcoplasmic reticulum was storing calcium ions. But if you open these little calcium gates by having the nerve impulse the muscle impulse shoot into the SR and cause gates to change shape and open, the calcium ions would leak out. When the calcium ions leak out, they have to have a place to go. And where they go is to the tropomyosin. The tropomyosin has troponin muscle chemicals in it, which are also proteins, and they attach to the calcium. So the calcium ions will come out of the SR, bond to the troponin on the tropomyosin, which is wrapped around the actin fibers in the sarcomere. When that happens, the tropomyosin changes its shape. Remember, the problem that the muscle cell had about contracting before was that it couldn't do it because the tropomyosin was in the way. But if we can change the shape of that tropomyosin and move it out of the way, get it away from the actin or wrapped around differently, that means that there might be an opportunity for the cross bridges to attach to the actin. And that's exactly what happens. The calcium ions come out of the sarcoplasmic reticulum, attach to the troponin molecules, and move the tropomyosin literally out of the way. It's like turning a switch on. When that happens, there's sites on the actin that attract the cross bridges, the ends of the cross bridges, and they attach to the actin. When they attach to the actin, they attach to the actin and spring to their relaxed state. In other words, just like a mousetrap, shooting its energy at the mouse. Boing, shoots, and then when that happens, it's pulling the actin closer together, and that makes each sarcomere a little bit shorter as the Z lines get closer. Now, here you see that actually happening. With the troponin moved out of the way, look at the configuration of the cross bridges on the top diagram. You see it pointing sort of to the edges. But once the tropomyosin moves out of the way, with the calcium ions on it. Look at this, at the configuration of those cross bridges. They are now pointing toward the center of the sarcomere. They have attached to the actin and literally pulled, just like a bunch of people pulling on a rope, and caused those two Z lines to come closer together, showing by the green arrows there. And you'll notice when the sarcomere is contracted, if you look at the diagram on the bottom left, you see that the sarcomere gets a little shorter. And because of that, each sarcomere does that in the muscle cell and the muscle myofibril, and many myofibrils do it. And then you have millions of cells. You got lots and lots of power, and you'll be able to pick up a heavy rock or even a suitcase or something like that if you needed to. So what's happening here again is that the tropomyosin attaches, the calcium attaches to the troponin on the tropomyosin, moves it out of the way. The cross bridges, which are already cocked back, they did that using ATP at the end of the last contraction. They pull it back at the end, just like you would set a mouse trap before you catch the mouse. And as soon as the cross bridge is attached to the actin, they do that power stroke, and then pull those, literally pull the two Z lines closer together on each sarcomere. Okay. Now, when you finish the contraction, you now want to relax the muscle. So relax the muscle, you have to get the calcium ions off of the tropomyosin so it can move back in the way. When it's back in the way, the cross bridges have to let go and the muscle relaxes. So you have to use energy, more ATP energy, to actively pump or transport the calcium back into the sarcoplasmic reticulum where it came from. As the calcium ions are released from the troponin, the tropomyosin moves back in the way of the myosin cross bridges and the binding sites. The cross bridges then use ATP to rearm themselves for the next contraction. In other words, as soon as it relaxes and the tropomyosin is in the way again and the calcium's back in the sarcoplasmic reticulum, ATP is used to cock back the cross bridges again, ready for the next time. It's like setting the mouse trap ahead of time. ATP is the energy source for this. Now, in muscles, you need so much ATP for this stuff because you're moving all the time, that there's a chemical in your muscles called creatine or creatine phosphate. And creatine phosphate is an energy molecule that could actually turn ADP and phosphate back into more ATP for your muscles. So your muscles, have a good supply of ATP to your contractions. This would work in smooth muscles and cardiac muscles as well. So this was the last slide. Now I'm going to do this. I'm going to review the whole thing step by step for you right now, and then we'll end the recording. Now, part one of this is the contraction phase. The nerve impulse reaches the muscle cell. Again, the nerve impulse is something you'll learn about next week, but today just think about it as a electric shock going through a wire. It goes very, very fast, hundreds of feet per second. The Golgi in the nerve cell at the terminal branches, the end of the nerve cell, releases acetylcholine into the gap, that little neuromuscular junction or synapse between the muscle cell and the nerve cell. The acetylcholine is called a neurotransmitter. Remember, Golgi is good at secretion, so the Golgi would release the acetylcholine into the gap. The acetylcholine diffuses across the neuromuscular junction gap, or the synapse gap, just spreads over there. It attaches to receptor proteins on the muscle cell membrane, which is also known as the sarcolemma. Sarcolemma is just another word for the muscle cell membrane itself around the muscle cell. It's just like a regular cell membrane made of phospholipids and proteins. When enough acetylcholine accumulates to meet the cell's threshold, in other words, there's enough there to get it started, kind of put like how much pressure you have to put on a trigger to have the gun fire. You put a little bit of pressure, it's not enough. You have to keep pulling until you finally reach that threshold, and then the gun will fire. Same thing happens here. When enough acetylcholine attaches to the muscle cell, it's over the threshold, and a muscle impulse has begun. Muscle impulse is exactly like a nerve impulse, and it shoots across the cell membrane of the muscle cell. However, in a muscle, it moves a lot slower than it does in a nerve, and next week we'll talk about why that is. The muscle impulse moves across the sarcolemma into the transverse tubules. the transverse tubules, which are also made of membrane, will take that impulse to the sarcoplasmic reticulum inside the cell. Inside the cell, there are gates on the sarcoplasmic reticulum that are affected by that impulse shooting through it, and they open and they allow calcium ions to leak out of the sarcoplasmic reticulum. The calcium ions attach to the tropomyosin, actually the troponin on the tropomyosin, changing its shape. When the tropomyosin is out of the way, myosin cross bridges can now attach to sites on the actin. As long as the calcium ions are present, the cross bridges do a series of power strokes and continuously cock back using ATP, pulling those two Z lines closer together, causing the muscle to contract. Now, when the muscle needs to relax, the calcium ions are removed from the tropomyosin and back into the sarcoplasmic reticulum using ATP. The cross bridges let go of the actin when tropomyosin moves back in the way. Again, if something's in the way, you let go. The muscle relaxes. But now the cross bridges are in the wrong configuration. They need to be cocked back again and get ready to boing for the next time. So they are cocked back or reset using more ATP. Now, very important thing. If this was the whole story, the muscle would contract and then contract again and then contract again and again and again and again and again until it dies. And that would be all over your body, and you would just fall into a ball and die. That's exactly what nerve gas does to you. Now, the reason for that is because we left the acetylcholine, which was the start of the muscle impulse, the trigger, on the muscle cell membrane. As long as that's still there, it's like you're holding back the trigger on a machine gun, and it just keeps firing, keeps sending another impulse to the SR, making the muscle contract again and again and again and again and again and again. And it would just keep doing that until you die. And it would be your intestines, smooth muscle, cardiac muscle, all the voluntary muscles. It would be a horrible way to die. Painful. That's what nerve gas does. And the reason that works is because there's another enzyme involved in the end of this story. And that enzyme is called acetylcholine esterase. And acetylcholine esterase is an enzyme on the muscle cell membrane that destroys the acetylcholine in less than a second, so that the acetylcholine only has a chance to cause the muscle cell to contract once. Once acetylcholine is destroyed, then you have to wait for more acetylcholine to come on, so you have to send another message down the nerve cell for it to happen. So you have to want to contract the muscle again. So without acetylcholine esterase, the muscle will just contract over and over again because the acetylcholine will cause that to happen on the muscle cell membrane. Many countries have nerve gas, and what that does, it destroys your acetylcholine esterase, so that when you contract the muscle, the acetylcholine is left on the membrane to start muscle cell contractions over again and over again and over again and over again until the muscle just basically poops out. Very painful, horrible thing. Is smooth muscles, cardiac muscles. Your heart will probably stop in the middle of the beat. You won't be able to breathe. You'll have incredible cramps. You'd be pooping and peeing and vomiting all at the same time probably, and all your voluntary muscles will be seem to be in a state of constant contraction, including your eyes and your tongue and everything else. It's a horrible way to die. Nerve gas is a terrible, terrible weapon, but it does work, and it works because it destroys the acetylcholine esterase enzyme, which is there to destroy the acetylcholine itself, so that it can only start one muscle impulse at a time. So anyway, that's the muscle cell story. Next week, we'll talk about the nerves and how that's all connected to the same story, especially what a nerve and a muscle impulse really is. So get your homework in on time. There'll be I think 2 questions on muscle cells for your assignment this week. That'll be due next Sunday. And, if you haven't got the others in, get them in on time. Okay, we'll see you next week, and have a good week. Bye-bye now.